This conference will now be recorded.
Hello everyone. Last day we ended with the topic types of peptide, and related to that, two questions we have discussed, two more questions we will also discuss. After that, there will be some test for amino acid, and you can also consider them test for peptide bond or protein, and then detailed structural relation informations for protein, and also, if time permits, denaturation of protein we will discuss today. So here we have this question, and it is saying that total number of negative charge in the tetrapeptide. What is the tetrapeptide? That is given GLY, then GLU, gly, glu, ASP, and then TYR. So we have total 4 amino acid connected to each other. So total 4 amino acids. So that is what is tetrapeptide. But peptide bond is total 3. It is at pH 12.5. Now pH 12.5 means it is basic pH. So the medium is basic. So under this condition, what will be the total number of negative charge? Now we know when we consider any amino acid, we mostly consider the zwitterionic structure, that is positively charged NH3 and CO2 minus. Carboxylic acid, it is not in CO2H form, it is in CO2 minus form. So negative charge, positive charge, both are present. But it is asking total number of negative charge and also basic medium, that you have to keep in mind, because there is acidic group, and if it is basic medium, so obviously it will remain in deprotonated state, and deprotonated state means there will be negative charge, but also there is positive charge. So we have to consider everything. But whatever we will do, we have to write the structure of the tetrapeptide, otherwise it is not possible to answer. So this is the structure of this tetrapeptide. So first we have, that is in the left hand side, which is the N terminal. So this side is N terminal, this side is C terminal. So that is the general convention we follow. So the first amino acid is GLY, which is glycine. So this is basically glycine. And then we have this peptide bond. The next amino acid is GLU, glutamic acid. In glutamic acid, this is the side chain CH2CH2 CO2H. And this is the peptide bond CONH. The third amino acid is aspartic acid, ASP. Now here the side chain is CH2COOH. This is the side chain for GLU. But now similar type of side chain, only one carbon is less. And the last amino acid, tyrosine, which is having this side chain. And as I have said that this is the carbon terminal. So that is why it must be ending with CO2H. You can also write the zwitterionic structure. That will also give you the same answer, but here zwitterionic structure is not written. Okay. Now when it is basic medium, you have to consider how many acidic groups are there. Fine. So here the acidic groups that are present: CO2H, CO2H, CO2H. So total 3 carboxylic acid groups are present. And there is also OH group present. Now OH, that is a hydrogen that is present in OH. That hydrogen is not so acidic compared to CO2H hydrogen, but look at this pH. It is highly basic. It is not just a pH 8 or 9, which you can say weakly basic. It is actually the extreme pH, close to 14. That is the extreme basic condition. So pH 12.5, that means it is very basic condition, and under that condition, this OH hydrogen, that will also be deprotonated. So there are total 3 CO2H, and all these H will be removed, and there is one OH group, so this H will also be removed. Fine. And if you consider zwitterionic structure rather than this type of structure, so you have to now write the structure like this. So in that condition also you will reach the same conclusion, okay? Because this NH3 plus, under this type of highly basic condition, it will not remain in the form of NH3 plus. Here this extra hydrogen, that will also be removed. So finally it will be converted to NH2. And 3 CO2 H group and one OH, all these will be deprotonated. So whether you write the zwitterionic structure or not, that doesn't matter. Ultimately, you will see there are total 4 negative charge. So 3 negative charges are coming from 3CO2H and the fourth one is coming from O negative. Okay. So this part is like phenol. So here it is written phenol will also lose its protons. So answer will be total 4 negative charge and no positive charge. Even if you consider the zwitterion structure, this positive charge will not be present because this extra hydrogen that will also be removed by this because of this basic medium in which it is kept, pH 12.5. This is integer type of question. Next one, that is also integer type of question. It is saying that in a linear tetrapeptide, again we have taken linear tetrapeptide just like the previous problem. Now this linear peptide, in the bracket what is mentioned, that it is constituted with different amino acids. So all the 4 amino acids are different, that is mentioned clearly in the question. Now you have to find, see, number of total amino acid minus number of peptide bond. So you have to subtract the number of peptide bond from the number of amino acid. So after subtracting, what will be the final value? That is actually we have to answer. So when it is tetrapeptide, obviously it means total 4 amino acid will be there. Amino acid I am writing in short like this, AA. And when there are 4 amino acid, obviously peptide bond will be 1 less. So if there are n number of amino acid, it will always be n minus 1 peptide. Okay. You can also consider the previous structure where all the 4 amino acids are different. And in this case also, if I highlight the peptide part, that is the peptide bond. This is the first peptide bond. This is the second one. This is the third one. So total how many? 1, 2, 3. But number of amino acid, total 4. So this is a particular example. So here the answer will be 4 minus 3. That means it will be 1. So this is 4. This is 3. If you subtract 4 minus 3, it will be 1. Okay, so this is the answer. Now we will see some test. So here the first test that we have, it is known as biuret test. And this test is also known as Piotrowski test. Okay. Now this is used, this test is used for detection of peptide bond. So you can say this is a test for peptide bond. So if you take a simple amino acid, that is only a single amino acid, it will not give test because at least one peptide bond must be there. So 2 amino acid must be present because for formation of one peptide bond there must be at least 2 amino acids, and that will be dipeptide. So this test will be positive if there is peptide bond. Now why the test is, we are calling it biuret test. What is biuret? Now this is the structure of biuret. See, NH2CO, then NHCO, NH2. So if you look at this molecule carefully, there is peptide type of linkage, NHCO, right? Now this molecule, biuret, in presence of copper ion, Cu2 plus ion. When biuret is treated with dilute copper sulfate solution, that means Cu2 plus ion, and it must be alkaline condition. Then this type of complex, so see, here we have total 4 biuret. This is 1 biuret, this is second, then third, then fourth. So total 4 biuret. The middle nitrogen, it will be coordinating with the copper. So total 4 coordination from 4 side, 4 biuret molecules are coordinating. And this complex, there is a specific color which is purple color. The formation of a chelate complex of copper, it is basically responsible for this type of color. And it appears, that is, it absorbs light in this particular wavelength. Okay. And according to wavelength, the color is, that will be changing also. So at this particular wavelength, the color that we will see, that is purple in nature. Now, this test, it will also be positive if you take any peptide, because for biuret molecule, there is also similar type of peptide bond. So because the bonds in biuret molecule, they are just, that is same, just like peptide bond. So that is why positive result for this test is obtained when the analyte, that is the sample, is containing any peptide. So this is the complex when there is a peptide. So see, copper is present as usual. Here we have this peptide bond and the chain is continuity because we have just taken a particular portion of a very, you can say very long peptide. So it may be penta or hexa like this. So CONH, this is one peptide bond, then another peptide bond, right? Then third one, and this is the fourth one. So similar type of structure as we have seen in case of biuret, and all these R groups, they are side chain. I mean, side chain that will be varying if amino acids are different. Here same R groups are written for simplicity, but that is not necessary. So what is required is that this type of presence of peptide bond must be there, and it is coordinating with copper 2+. So before reaction, that is, if you take simple copper sulfate, the solution is blue in color. So this is that is, if you are adding this copper sulfate to some analyte or sample where peptide bond is not present, so obviously there will be no change in color. So that is why the color will still remain same. This is negative taste. But if there is presence of peptide bond, now the color will change from this type of blue to purple, and it indicates that the taste is positive. Okay, so in this way, from the color change, we can identify whether peptide bond is present in the sample or analyte or not. Okay, this test is done for aromatic amino acid, not just any amino acid, aromatic amino acid. Now when I'm saying aromatic amino acid, that means the side chain should contain any benzene ring, and this reaction is known as xanthoprotic test. So this is used to detect amino acid which is containing any aromatic nucleus. For example, tyrosine, where aromatic nucleus is present in the side chain. Tryptophan, there also indole moiety is present. Then phenylalanine, though for phenylalanine there is some special discussion we will do. But remember, for aromatic nucleus, if it is present, you can also consider protein solution. Though protein, what is exactly, that we will discuss after finishing all these tests. But protein you can consider, that is one type of polypeptide where several peptide linkages are present. Okay, so in this case also, that is in protein solution, if there is any side chain which is containing aromatic nucleus, then this test will be positive. So it gives yellow color nitro derivative when heated with concentrated HNO3, nitric acid. So you will take the sample, you will heat it with concentrated nitric acid, and there will be formation of nitro, that is nitro group will be inserted and the color will be changing. Now aromatic benzene ring, basically it will undergo nitration. Remember, simple nitration reaction for benzene. So here also same thing will be there, and the product will be yellow. So that is why from color change we can understand that something is happening. Okay, so see here we have taken tyrosine where the side chain is CH2, then pH ring, then fourth position OH. So this is aromatic in presence of concentrated HNO3 in this aromatic ring. With respect to OH, if you consider the ortho position, NO2 group is inserted. So this is as if electrophilic substitution, where the electrophile, that is E plus, is basically NO2 plus. So NO2 plus, it is inserted in the benzene ring. Hydrogen which is present at ortho position with respect to OH, that hydrogen is replaced by NO2 group. So this is nitration of the benzene ring. Now this test will be positive because of this insertion of NO2 group. Similarly, if you take tryptophan where we have CH2 and then indole moiety, but again aromatic ring is present, and here nitration will be at this position, but color will be changing. But when it is phenylalanine, now what happens? Mostly it gives negative taste, or sometimes rarely weakly positive reaction is possible. That means very few amount of nitro derivative will be produced. Though this amino acid also contains aromatic nucleus, but here we can see some exceptional result, because under the same condition it is difficult to nitrate this molecule. Okay, so specifically for tyrosine and tryptophan, if these type of side chains are present, this test will be positive. And further addition of alkali, that is after formation of this nitro compound, if you further that is if you do some one step extra, which is addition of alkali, then the color will be deepening, it will be changing from yellow to orange. Okay, so see, this is the color change. If the test is not there is no aromatic nucleus present in side chain in the sample, it will be negative, there is no yellow color, and it will be yellow when nitro group is inserted, and if you add alkali, it will be changing to, it will be more intense, it will be changing to orange. So in this way we can understand whether not just amino acid, aromatic amino acid, if aromatic ring is not present in the side chain, it will not be positive. So it is specifically for tyrosine and tryptophan, because these two are the mostly aromatic side chain we see, because another important aromatic side chain amino acid phenylalanine, but here the test is mostly negative. Okay, now the third test, which is very common, and this test is done to identify whether there is any amino acid present or not. So basically not just amino acid, this test is you can do for any primary secondary amine, and we know when it is amino acid there is presence of primary amine group, so it is positive for any primary or secondary amine. So this is primary, this is secondary, and as we know in amino acid primary NH2 group is present, so it will also give positive test, even it is positive for ammonia. Fine. So in this test what happens, we use a reagent which is known as ninhydrin. Obviously we'll see the structure, that is added to the sample or analyte, and what happens here, deep blue color generation is there. No, though it is not always true that you will get deep blue color, though that is the most common color, but in some cases there may be a little bit different types of color, only in some cases. And this deep blue color, that is also known as Ruhemann's purple. So this is the reaction. This is amino acid general structure we have taken, where R is the side chain, and this is the structure of ninhydrin, which is the actual reagent we are going to use. So in this case what happens, if you look at the amino acid carefully, this RCH part, as if this part is removed in the form of aldehyde, then NH2, now it is removed in the form of ammonia, and CO2H, it is removed as CO2H, and one of this OH that you can see in ninhydrin structure, now in that OH we have H. So one oxygen is extra in this position. So ninhydrin, one OH is removed and one oxygen we are getting. So amino acid, now its fate is, it is converted to aldehyde, CO2, ammonia, these are the byproducts, and this is reduced ninhydrin. Okay, so as if ninhydrin is the oxidizing agent here, because it is itself reduced. Now this is the first step. In the second step what happens, now this ninhydrin, there is nothing further for amino acid, it is already converted to aldehyde, but this reduced ninhydrin, now it can react with this ammonia, which is the byproduct in the first step, and the second molecule of ninhydrin. This is the reduced ninhydrin, this is original ninhydrin molecule, and ammonia. These three together, it will make this type of complex, which is violet blue color, that is this Ruhemann's purple. But as I have said, in some cases you can have different types of color. So if we interpret the result, for ammonia, primary, secondary amine, any amine, and amino acid obviously, deep purple color is obtained. If it is hydroxyproline, remember hydroxyproline or proline, which is another type of amino acid where the side chain is containing, there is a ring type of structure, it is secondary amine. So for proline and hydroxyproline, you will not get deep purple color, but what you are getting, you will be getting yellow color. Then for asparagine, which is another amino acid, brown color is obtained. And if you are seeing no color change, that means there is no presence of amino acid or amine, primary, secondary, whatever, or ammonia. So this is the test which is mainly for amino acid. It is not for peptide bond or protein, and not just amino acid. It is also positive for any other amine and even for ammonia. Okay. So now we will go into the details information about proteins and also structural information. We know what is amino acid. This is the basic unit. And then different amino acid, when they are connected to each other by peptide bond, we get peptide. Now this peptide, depending on how many amino acids are involved, it may be di, tetra, penta, etc. But when this peptide, it is containing very high number of amino acid, now it will be protein. So both polypeptide and proteins, they are organic compounds that are composed of amino acid. So if peptide and protein, if you are considering them as polymer, then the monomer unit is amino acid. So these are the polymers which are made out of amino acid acting as monomer. So amino acids, they bind together to form polypeptide chains, and these chains, when this is very long, they can combine to form a protein. Now proteins, they are very large and complex. That is the difference with peptide. Peptide is just like protein, but the difference is protein is very large, very complex. So many different types of amino acids are there, and molecular weight is very high. And for protein, when we will see the structural information, then it is clear that 4 levels of arrangement of proteins are there: primary, secondary, tertiary, and quaternary. So structure of protein. Protein structure can be described at 4 hierarchical level. Hierarchical level means first it will be primary, which is the simple way, that is the simple, I mean to say, among these 4 is the simplest structural information. Then the next higher position is secondary, and in this way tertiary and quaternary. And when you will move from primary to secondary, it will be more complex, and then secondary, tertiary, again more complex, and finally quaternary. This is the most complex. Okay. Now primary, which is very simple, we know that proteins are polypeptide chains made up of amino acid and that are connected to each other by peptide bond, that is CONH bond. Now this relative arrangement, or you can say sequence of the amino acid. Okay. So sequence of amino acid means which amino acid is coming after which amino acid. There may be some sequence. So this sequence is actually the primary structure of protein. And knowledge of this, it is important, because if you just change one amino acid, okay, suppose we have, just for simple example, we have glycine, then we have GLU, then we have ASP, like this. Now suppose if you just change one amino acid, GLU, with some other amino acid, suppose you are changing it with serine amino acid, so obviously GLY, GLU is ASP, and GLY, AC or ASP, these two are not same, though this is very simple, this is tetrapeptide. But suppose if you take protein which is very large, then this type of possibility is very high. That is, you are just changing one amino acid in between and the complete properties change. So that is why even small changes have the potential to alter the overall structure and also the function of the protein. So that is why primary structure, which is nothing but the sequence of amino acid, this is also very important. So here we have taken this polypeptide chain. You can also consider it as a protein, and these each small dots, if you enlarge, suppose it is phenylalanine, then leucine, serine, cysteine. So in this small part, suppose if you are changing any one of these, suppose you are changing this one, so obviously there will be effect. Fine. So this is the primary structure, the most basic, most simple structure. Now after this, the next one is secondary structure. So it refers to conformational relationship of nearest neighbor amino acid with respect to each other. So sequence of amino acid is primary. Now conformational relationship, that means if we have, I am writing this polypeptide where we have different amino acid. Okay. Now suppose this is amino acid number 2, this is 1, 2, 3, 4, and this is 5. Now what is the relationship conformation? So relationship of 1 with 4 or 1 with 5. How, that is whether they are close to each other or not. All this information we can get in secondary structure, and that is conformation relationship, that is nearest neighbor amino acid, how they are placed with each compared to each other. Now in this case hydrogen bonding occurs, and it occurs between an NH group of one amino acid residue. Fine, NH means the NH group which is present in peptide bond NHCO. So NH of one amino acid residue and the carbonyl oxygen of another amino acid residue which is properly situated, and the second amino acid residue which is neighbor of the first amino acid residue. So suppose it is at position 1 or 5 or 1 or 6 like this. So there will be hydrogen bonding, and this hydrogen bonding is actually responsible for the secondary structure. So when there is hydrogen bonding, they will be coming close to each other. Okay, so that is why this is the deciding factor of the secondary structure, the hydrogen bonding of NH group of one amino acid and the carbonyl oxygen of another properly situated amino acid, not just any amino acid, another properly situated amino acid. And two most common substructure will be there, that is two types of secondary structure possible. One is alpha helix. So as you can understand, helix means helix type of structure, spiral structure will be there. Another one is beta pleated sheet, which is plate like structure. So in a simple way, if you consider secondary structure, so this part is basically alpha helix. Fine. So alpha helix, this part if you focus, not the other part is beta sheet where plate type of structure is there, and all this linkage that you are seeing, that is actually hydrogen bond. So suppose here we have NH bond and there is C double bond O. So hydrogen bond is possible between hydrogen and oxygen. Okay, and that is, as there is hydrogen bond, so that is why there is this type of spiral type of nature. Similarly, when it is beta sheet, now the hydrogen bonding, it is not like alpha helix, but still there is bond, and that is why they are coming close to each other, but it is plate like structure. Then again in this part you can see alpha helix structure. So these two are the possibilities when we are considering secondary structure. Next, we will go into the details of alpha helix and also details of beta pleated sheet. So for alpha helix, so this structure, as you are seeing here, mixture of both are present. Okay, so mixture of both are present. First, we will enlarge this part. So this part, as you can see, this is spiral in nature, right? Now this spiral in nature, the part that you are seeing, if you enlarge this part, now there will be something like this. Okay, so see here NH, this H of this peptide bond, now it is interacting with the carbonyl oxygen of another properly situated amino acid. So this is the hydrogen bond. In the alpha helix substructure, the amino acids are arranged in a right-handed helical or spiral structure, and that is formed, that is this type of structure, which factor is responsible? Hydrogen bond between carbonyl oxygen of one amino acid, which you can consider as if nth residue, and the amino hydrogen of the fifth residue. So if it is N, then the next amino acid that will be at, that is compared to its position is fifth, or you can say N plus fourth residue. The side chain of the residues, they will protrude outside the helix. So see here, though side chains we cannot see properly, but CONH, if this is the peptide bond, then this will be the side chain. Then Na again NHCO, this will be the side chain. Then NHCO, this will be the side chain. So side chains, they will not be towards, that is in the this spiral part, but they will be protruding outside the helix. Each turn of this alpha helix contain about 3.6 residue. So each of these turn that you are saying, one complete turn, there will be total, not exact 4 amino acid, it is fraction. In this one complete turn, there will be 3.6. So remember this value, you may get question regarding this. And what is the length of this one complete turn? It is 5.4 angstrom. So these two values, and also this information, that is the NH hydrogen bond is formed between hydrogen of one peptide NH hydrogen, and the next one is at N plus 4th position. Okay, so these three informations are important. Now when it is beta pleated sheet, now focus on this part. So this part is not spiral, it is plate-like structure. This structure, all peptide chains are stretched out to nearly maximum extension, then laid side by side, which are held together by intermolecular hydrogen bond. So here also hydrogen bond is present, but it is not like alpha helix hydrogen bond, which is basically formed between nth peptide bond, and then the next one is N plus 4th peptide bond. Here it is not like that, but hydrogen bond is such that ultimately you get plate type of structure. So if we enlarge this part, you can see this is pleated structure. Fine. And you can also see the hydrogen bond. So suppose this is one sheet, then next sheet, then the next sheet, and each sheet, how they are connected to each other. That is, if you considering 3 sheets, as if these are the hydrogen bond like this. Okay. So that is all about alpha helix and beta pleated sheet. After secondary, if we consider in hierarchical position, the next complex structure is tertiary. So tertiary structure, it is basically the overall three-dimensional structure of the polypeptide chain that is present in protein. In case of secondary structure, you can say that is also 3D, but it is not overall 3D arrangement. It is the conformational relationship of the neighbor amino acids. But in this case, tertiary, it is more complex. You have to now consider the structure in a broader way. Now it is overall three-dimensional arrangement. That is, after secondary structure, what is the further folding possible? Further folding of the secondary structure. So the way the chain is folded, it affects both the physical properties of a protein and also the biological functions that it is offering. So that is why tertiary structure, it is important as for to know. And the two most important shapes for tertiary structure, which is globular or spherical, and fibrous or rod-like. Now sometimes these two types of proteins, it is also considered under the heading of classification of protein, that is depending on the based on the overall three-dimensional arrangement, what are the different classification possible, then these are the two types, but you can also consider them as if it is under tertiary. Now this tertiary structure will come to in details of this fibrous and globular, but before that we have to know that in tertiary structure, when you are getting further folding of the secondary structure, what are the factors that are responsible for this further folding? So tertiary structure of proteins, they are stabilized by different interactions between the side chains of the amino acid. So here you can see total 4 types of interactions are there. Hydrogen bond, that we have already seen, but some more interactions are possible. Suppose if there is cysteine residue, remember cysteine residue where sulfur SH bond is present, and in this case disulfide bridge is possible. So SH group of one amino acid residue, another SH, if these two are connected, now there will be formation of disulfide linkage like this. So as if two amino acid and the SH group present in side chain, now they are connected to each other, that means they will come close to each other. So this type of disulfide bridge is possible, but it is only possible when there is cysteine side chain, otherwise it is not possible. Then electrostatic or hydrophobic interaction. Electrostatic interaction means interaction between positive charge, negative charge, or it may be hydrophobic also. And van der Waals interaction. Now all these interactions, if we see, Van der Waals interaction, it is mainly non-polar interaction. If there is any non-polar group, then Van der Waals interaction is possible. So see, all these interactions you can see in this single picture. Hydrogen bond, that is very common. So suppose you have any CH2 CO2H type of residue. Another residue is CH2OH. So hydrogen bond is possible between hydrogen and oxygen. Then hydrophobic interaction. Look, focus on this part. Here we have this side chain which is CHMe2. Another side chain is also same. So these two are hydrocarbon part. So this part, they are hydrophobic. That means they will not be attracted towards hydro. That means non-polar. So towards polarity they will not be attracted. Okay. So there will be hydrophobic interaction which is like this. Then disulfide linkage, we have already seen it. And ionic bond. So positive in charge, negative charge. So electrostatic interaction is also possible. And Van der Waals interaction, it is basically I just omit this part. Van der Waals interaction, that is one type of hydrophobic interaction, because when there is non-polar group, just you are seeing here CHMe2 or any other alkyl group, then this type of interaction is possible. So total 4 interaction. Now these globular and fibrous, we'll see the details. So you can consider this discussion as if it is classification of protein. So proteins are classified based on their overall shape into 2 major types, fibrous and globular, and this falls under tertiary structure of protein. So as the name suggests, when it is fibrous, it will be rod-like structure. So see the overall structure, it is rod-like structure. They are linear molecules just like fiber. They are insoluble in water, held together by disulfide bridge and weak intermolecular hydrogen bond. 4 interactions you have already seen there. And among these 4, mainly these 2 interactions are observed here. They are insoluble in water, important property. And this type of proteins, where you will find, it is mainly responsible for structural, that is structural protein, I mean. So for example, keratin, collagen, keratin, you know, it is present in here. So it gives some particular structure. So that is why they are structural protein, and it is water insoluble. When it is globular, now the overall shape, it is spherical type of shape. That is why it is globular, because here the polypeptide chain, they are folded in such a way. So ultimately you get some globular or spherical type of structure. One important difference with fibrous is that now they are soluble in water, and their functions are completely different. Their functions are maintenance and regulation of the life processes, and this requires mobility. And when it is about mobility, obviously water solubility is important, because if it is not water soluble, because mostly the reaction inside our body, that is in vivo, it is occurring in aqueous medium. Okay. So if it is not water soluble, how it will be mobile? These are the examples. But when it is structural protein, mobility is not required. So that is why in this case, if mobility is present, there will be problem. So that is why it is water insoluble. But globular protein is water soluble. So these are some important difference between fibrous protein and globular protein. And this is under tertiary structure of protein. And after tertiary, finally the quaternary structure, and this is the ultimate highest complexity we have reached. Some proteins are made up of more than one polypeptide chain. Now what will happen? If there is more than one polypeptide chain, these individual polypeptide chain, how they are interacting with each other, that is actually the quaternary structure. One very common example that is taken mostly is hemoglobin. So in hemoglobin, which is basically oxygen transporting protein, there are total 4 polypeptide chains. Then again, another example, which is DNA polymerase. So one example is hemoglobin, second example is DNA polymerase enzyme, which is also protein. What is enzyme? It is nothing but biological catalyst. Detailed discussion separately we will do. So DNA polymerase enzyme, just remember this is also a type of protein, and its function is to make copies of DNA. So DNA polymerase enzyme, which is also a protein, in this case, total 10 polypeptide chains are present. So see, it is even more complex. So now we will take the simplest among these two, which is hemoglobin, containing 4 polypeptide chains, and all these 4 polypeptide chains, you can see it is shown here by different colors. So this is one, then second, third, and fourth. Now among these 4, it is how it is called. 2 polypeptide chains are called alpha chain. So see, this is alpha, the red one and the purple one, which is containing total 141 amino acid residue. And there are also 2 beta chains. So this is beta, the pink one, and the green one, that is also beta, which is containing 146 amino acid. But there are total 4 unit, that is 4 polypeptide chain, and hemoglobin is present in red blood cell, that we know. So in this case, their relative arrangement with respect to each other, that is actually the quaternary structure. So in these proteins, the individual polypeptide chains, which you can also call as subunit, they interact with each other to form the multimeric structure, and that is actually the quaternary structure. Now, what are the interactions that are responsible for this type of structure? That is actually same as tertiary structure. So whatever interaction you have seen in case of tertiary, remember van der Waals, hydrogen bond, disulfide, all these are also present in this case. But it is only applicable for those proteins where it is more than one polypeptide. So each polypeptide chains, how they are interacting with each other, that is actually the quaternary structure. So now we have discussed all the 4 structure, and if you see all these structure simultaneously in one place, it will be something like this. So this is a primary structure, that is a sequence of amino acid, and gradually if you move to the next hierarchical position, this is secondary structure. Now in secondary structure we know alpha helix and beta plate. So as if this part is beta, which is plate-like, this is spiral, which is alpha, and these are the substructure alpha and beta. Then tertiary structure, overall 3-dimensional arrangement, and for quaternary structure, that is possible when there are more than one polypeptide chains. Okay, so all these 4 different type of structure you can see simultaneously in this slide. Denaturation of protein. So as the name suggests, denaturation, that means there will be some change in the structure of protein. We know that we have just discussed that in biological system, protein is found with unique 3-dimensional structure, and also their biological activity is fixed, right? And it is when they are present in their normal state, we will call them native protein. And this is very unique for each protein. Now this unique 3-dimensional structure, that is formed by 4 interactions that we have seen: disulfide, hydrogen bond, hydrophobic or van der Waals, and electrostatic interaction. But suppose if for some reason these interactions are destroyed, then what will happen? So when a protein, it is in native form and you are making some change, it is subject to some physical change, and that is possible if you are increasing the temperature, or sometimes there may be even chemical change. How that is possible? Suppose you are if you are adding some chemicals such as urea. Urea is NH2CO NH2. This is urea structure. Or you are doing some alternation of pH. Suppose you are changing the pH drastically from acidic to basic. Or if you are changing the ionic strength of the medium in which it is kept, then there will be some chemical changes also. But whether it is physical or chemical, in both cases there is this 3-dimensional structure, its uniqueness is lost. It leads to the loss of 3-dimensional structure, either partially or completely. Okay, so very common example, it is egg, and when we are increasing the heat, it is changing its form. Now it is solid, it is becoming solid. Okay, so this is thermal denaturation. This is physical change, higher temperature. Fine. Then if you add a little amount of curd in warm milk, it will, and if you keep it for some time, it will be converted to curd. So in this case, we are increasing the acidity, and there is a specific bacteria which is basically doing this. But here chemical changes occur. Okay, alternation of pH. So 3D structure will be damaged. So this is denaturation. The process of losing its higher order structure without losing the primary structure. Remember, here three-dimensional structure is lost, but no effect on the primary structure. Because if you want to destroy the primary structure, basically you have to break this peptide bond, CONH bond, and this peptide bond, it is not so easy to break. Okay, so since denaturation reactions are not strong enough to break the peptide bond, so primary structure will not be, it will remain intact, but other structure will be disturbed, and this is known as denaturation. This is important information. You may get question like this, that when denaturation is occurring, which structure is actually there is no effect, and the answer is it is primary structure where there is no effect. And when in a simple picture, you can consider this. This is native protein. There is some specific uniqueness in the structure. This is denatured protein. And when it is denatured, biological function will be completely lost, because biological function is related to the unique three-dimensional structure. But if that uniqueness is already lost, so obviously biology function will also be affected. Okay. So enzyme, remember one this term we have used, DNA polymerase enzyme. So enzyme is nothing but protein. But why we are discussing it separately? Because they are doing some specific work. There are many biochemical reactions that are occurring in the living cell in vivo. And example is digestion of food, absorption of particular molecules from the food, and ultimately it will be converted to energy. So that is the purpose, that is through diet how we are getting energy. So different reactions are involved. It is not very simple process. It is a complex process, right? So in this process, all these reactions are catalyzed by some special proteins. So enzymes are nothing but proteins, but they are special proteins where their job is to catalyze. So you know what is catalyst? In vitro, that is outside our body. When we do some chemical reaction, we use some catalyst. For example, when we do hydrogenation, metal catalyst hydrogenation, we use platinum, nickel, this type of catalyst. But suppose this reaction which is occurring inside our body, that is biological reactions, they are also catalyst required, and this natural catalyst, that is a biological catalyst, that is actually the enzyme. So these biocatalysts, they can accelerate the reaction rate, because that is the definition of catalyst, general definition. Why we use catalyst? Because it is such a thing which when we add it to some reaction in very little amount, there is no change in its own structure, but reaction rate will be highly increased. So here also you can see the reaction rate, now it is increased 10 to the power 5 times. And also these catalysts are very specific. So you can use a particular enzyme only for a particular reaction, not just for any reaction. Okay, so that is why it is specific. Some characteristic of enzyme catalysis reaction. A single molecule of enzyme catalyst, they can transform up to 1,000,000 molecules of the reactant per second. So you can understand how that is, if their efficiency is very high. Just one enzyme molecule, it can help in the conversion of reactant to product. How many? Million. Okay, so this is very efficient. Then these biochemical catalysts, they are very unique to certain type of reaction. So that is actually this last point. They are highly specific. Same catalyst you cannot use in more than one reaction. So here we have two examples. The example one, carbonic anhydrase enzyme. So this is a name of a particular enzyme, and from the name of the enzyme also you can understand its role. Carbonic, so it is involved in the conversion of carbonic acid to water and CO2. What is carbonic acid? Carbonic acid is H2CO3. So depending on what type of reactant we are using, there is some, you can understand the name is based on that, carbonic anhydrous. It is helping in the interconversion of carbonic acid to water and CO2. So this is specific for this reaction. Then another catalyst which is known as sucrase. This enzyme is catalyzing the hydrolysis of sucrose to fructose and glucose. You know, when we do hydrolysis for sucrose that we have discussed in carbohydrate part in this chapter only, we have seen that it is converted to fructose and glucose. So this reaction is catalyzed by the enzyme sucrase. So here also you can see there is similarity in the name. Okay, so that is, it is so specific. The effectiveness of enzyme, it is maximum not just at any temperature, there is an optimum temperature, and also there is optimum pH level, and mostly when it is inside the body, this range is from 5 to 7. So it is not very acidic, it is not very basic, it is more or less neutral pH. Okay. Mechanism of enzyme action. Enzymes are biocatalysts. So they are also catalysts, but we will call them biocatalysts because they are used inside the body in vivo. They catalyze specific biochemical reaction. They generally activate a reaction by reducing activation energy by stabilizing transition state, and that is the general property of any catalyst, not just for enzyme. So suppose if we consider any energy profile diagram, if this is the that is energy and this side is reaction progress, so as you move from left to right, reaction is progressing, and in this case this is reactant, this is product. So if I draw a line, this is the activation energy. Okay, but when you are using catalyst, now what happens, it stabilizes the transition state. This is the transition state energy. Now when you are using catalyst, now the energy profile diagram is something like this. So now this is the new transition state. Suppose this is 1, this is 2. So this new transition state, now if you take this gap, it is lesser than the previous one, and this gap is actually the activation energy. So in this way, when we are using enzyme, activation energy is reduced, and when activation energy is reduced, obviously reaction rate will be increased. Enzyme, how they are actually doing this job, they consist of a number of cavities, and these cavities, there is a particular shape. This is known as active site, and it is present on the outer surface of this. Now the enzyme E and the substrate, that is a reactant, what happens, the enzyme, it will attract the substrate not just at any particular position, it will attract the reactant or substrate S to its active site only, and it will produce intermediate enzyme substrate complex, which is known as ES. So look at this structure. This is enzyme in blue color. These are the active site which is having some specific shape, and this shape must be complementary to the shape of substrate. So here we have taken sucrose, which is the substrate here. Remember which enzyme is used for hydrolysis of sucrose? Sucrase enzyme. So this enzyme is sucrase. Now this will be attracted towards this, and there will be enzyme substrate complex, and the shapes are complementary to each other, so they are fitting to each other completely, and this ES, that is enzyme substrate complex, ultimately it will be converted to, that is it will dissociate to product. So to first it will produce enzyme substrate complex, catalyze the chemical reaction by which products are formed, and finally it will be dissociated to product, that is enzyme, it will be regenerated. So these are the products, that is one part is glucose, another one is fructose, and the enzyme also that will be regenerated. Okay, now this type of mechanism, that is the shapes are complementary, they are fitting to each other properly. So that is the reason each enzyme is specific for each substrate. You cannot use sucrase enzyme for interconversion of carbonic acid to water and CO2. Okay, then you will need another catalyst, enzyme. Now this mechanism is known as lock and key mechanism. as if the enzyme is key and this is or you can consider this is enzyme and this is substance, whatever it is. So you have to understand that here this is the correct key, this is the incorrect key. Now why I am saying so? Because if you look at the shape of the enzyme, then this part there is no difference. But if you look at this part, so this green color part, that is actually the empty part where the key will fit. So it is completely matching with this. Now when the key is inserted, now it is matching. But if there is no such matching, it cannot fit properly. So key cannot be inserted, right? So similarly, if you take incorrect key, now this part, the shape is not matching. It is like a V shape, but here it is straight. So there will be still some gap. Okay. So this is not fitting properly. So no reaction will be possible. It is correct fit, so reaction is possible. So this is lock and key mechanism, which is nothing but proper fitting mechanism, and that is actually the reason why uh, enzymes are so specific in their reactions. Fine. So one problem regarding enzyme, we have to identify the correct statement regarding enzyme. The first statement, enzymes are specific biological catalysts that cannot be poisoned. And what is poison? That we will see. Normally heterogeneous catalysts that are very specific in their reaction. Now specific, it is true, but whether they are heterogeneous or not, that we have to check. Then there are specific biological catalysts that can normally function at very high temperature. Now this is obviously not correct because it is inside our body there is not high temperature levels. Okay, so this is not possible. And enzymes are specific biological catalysts that possess no well-defined active site. Now this is not at all true. So this you can completely reject. Now in this two, if we are stuck, now we have to see what is poison. So catalyst poisons are sometimes used for catalysts which inhibit or destroy the activity of catalyst. But inside the body in vivo, this is not possible. So enzymes are those catalysts which cannot be poisoned. So this is actually true, and we have to find the correct state. So this is actually should be the correct answer. Then it is not necessarily it has to be heterogeneous. It may be heterogeneous, it may not be. Okay. So cellular medium catalysts are homogeneous, but membrane bound are heterogeneous. So it may vary. It may be both. So this is also not completely correct. And this is the temperature which is optimum for catalyst, not 1000 Kelvin. So considering all these, this should be the correct statement. Option 1 is the correct option. So we will end here. We are at the end of the session. Thank you for listening.